Hello and welcome back to Cellular and Molecular Biology, or BIOL1414. This is your Chapter 13 PowerPoint. This PowerPoint comes from Module 3 of your Openstax book, Biology 2E, and Chapter 13 covers chromosomal theory and genetic linkage. Starting off with our objectives. Objective 1 is we want to be able to understand chromosomal theory and genetic linkage. Objective 2 is to be able to understand the chromosomal basis of inherited disorders. And number 3 is to recognize some vocabulary terms, including karyotypes, non-disjunction, aneuploidy, duplications, deletions, and translocations. So let's start off by talking about the chromosomal theory of inheritance. So the observations for this theory were originally made by Theodore Boveri and Walter Sutton in 1902. Theodore Boveri observed that proper sea urchin embryonic development did not occur unless chromosomes were present. And meanwhile, that same year, Walter Sutton observed that chromosome separation into daughter cells during the process of meiosis was possible. 10 years after the chromosomal theory of inheritance was proposed by Boveri and Sutton, Eleanor Carruthers observed independent chromosome assortment in grasshoppers, officially discovering physical evidence that supports chromosomal theory of inheritance. So once again, chromosomal theory of inheritance identifies chromosomes as the genetic material that's responsible for Mendelian inheritance, the process of passing down genetic material. So now let's talk about the process of homologous recombination, which is also referred to as crossing over. So alleles become unlinked and chromosomes physically exchange segments. This allows recombination of DNA and contributes highly to genetic diversity. So looking at the image here, I always like to compare this to the chromosomes basically holding hands. So if you're looking here at the far left side, you have homologous chromosomes that are aligned, homologous meaning one. So if you notice, each individual chromosome is all one color. The one on the far left is blue, and then here you have a red. In the next portion, you'll notice that crossing over is starting to occur right here. So this is where the blue and red chromosome hold hands, and then by the third phase, the recombinant chromatids have formed, which means that this chromatid here and then this chromatid here have, as you can see, exchanged some genetic information with one another. You still have two non-recombinant chromatids, but this is the reason that we have so much genetic diversity. We now know that the pairing and interaction between these chromosomes, or what we call synapsis, does more than simply organize the homologs for migration to separate to daughter cells. This really does contribute highly to genetic diversity overall. I'd like to briefly talk about genetic maps. So due to the limitations of the time, when things were discovered back in 1902 and then subsequently proved in 1912, due to those limitations, scientists were not able to prove that crossing over was taking place, and it was not widely believed. Many scientists did not understand how chromosomes could possibly break apart and then rejoin in that way. We now know that homologous chromosomes exchange segments in meiosis by reciprocally breaking down and rejoining their DNA at precise locations. So there's something called a genetic or chromosome map, and it's a linear representation of gene order and relative distance on a chromosome. The average number of crossovers between two alleles, basically their recombination frequency, correlates with the genetic distance from each other relative to locations of other genes on that same chromosome. So now let's talk about chromosome identification. And this is the point in the slideshow where we're going to start talking about some of those vocabulary terms that we talked initially about in the objectives portion. And we're going to start with karyotype. So a karyotype is the number and appearance of chromosomes and includes their length, their banding pattern, and their centromere position. To obtain a view of an individual's karyotype, cytologists can photograph the chromosomes, and then they cut and paste each chromosome into a chart, which we call a karyogram. In a human karyotype, autosomes, or what we consider body chromosomes, anything that's not a sex chromosome, they're generally organized in approximate order of size from the largest chromosome being 1 to the smallest being 22. The X and Y chromosomes are not autosomes. However, often chromosome 21 is actually shorter than 22. It's also possible to identify chromosomes by their number, size, centromere position, and banding pattern. And once again, in humans, we label our chromosomes from the largest on the left side of our karyogram to the smallest on the right side. X and Y chromosomes, once again, are not autosomes. Autosome meaning self-some, auto meaning body chromosomes. So now let's talk about chromosome number disorders. So once again, hitting on a vocabulary term, non-disjunction occurs when homologous chromosome pairs, or those that look alike and are together, or sister chromatids, fail to separate during meiosis. The risk of non-disjunction does increase with the parent's age, and typically we see this with an increase in the mother's age as the eggs age. Non-disjunction can occur during either meiosis 1 or meiosis 2. The result of non-disjunction is an abnormal chromosome number, leading to an individual who is considered aneuploidy. That means an individual with an error in chromosome number. So the most prevalent or most widely known non-disjunction is Down syndrome, or what we call trisomy 21. It's the most common, and it occurs at about 1 per 800 births a year in the United States. One of the most common genetic birth defects, and according to the National Down Syndrome Society, there are more than 400,000 individuals with Down syndrome in the US. Patients with Down syndrome have 3 copies of their 21 chromosomes instead of the normal 2. The major clinical features of Down syndrome patients include low muscle tone, small stature, and upward slant to the eyes, a single deep crease across the center of the palm, mental retardation and physical abnormalities, including heart and intestinal defects, and increased risk of leukemia. Every person with Down syndrome is a unique individual and may possess these characteristics to different degrees. And this, once again, is a form of non-disjunction leading to a person that we consider aneuploidy with an error in their chromosomal number. So let's talk a little bit more about chromosome number disorders. An individual considered euploid, or EUPLOID, is an individual with what we consider the appropriate number of chromosomes for their species. Once again, someone considered aneuploid, that's an individual with an error in chromosome number. Aneuploid is a term that includes mesonomy, which is losing one chromosome, or trisomy, gaining an extraneous chromosome. So once again, Down syndrome being chromosome 21, the most common example of trisomy. It's often called trisomy 21. You can also have an individual who is polyploid. This is an individual with more than the correct number of chromosome sets, 2 for diploid species. For example, fertilizing an abnormal diploid egg with a normal haploid sperm would yield what we call a triploid zygote. A chromosomal segment can duplicate or delete itself. So duplications and deletions often produce offspring that survive, but do exhibit some abnormalities. So today we've learned that chromosomal structure is not always uniform. Chromosomes can undergo rearrangements. So for chromosomes, there are 2 possible options that we'll talk about today, one of them being inversions and the other being translocations. These are the 2 most common structural rearrangements that take place in chromosomes. So inversions only change gene orientation, and they're not likely to have as much of an effect on an individual as an aneuploid error if they don't disrupt a gene sequence. But with translocation, which is what this image represents, it occurs when a chromosome segment dissociates, basically detaches, and then reattaches to a different non-homologous chromosome. So there is no gain or loss of genetic information here. The portion of the chromosome that's separated is genuinely just moving over after translocation. It's on a different chromosome altogether. So there's no gain and there's no loss of genetic information. It's a reciprocal translocation, and it occurs when a DNA segment transfers from one chromosome to another, non-homologous chromosome. So this wraps up our Chapter 13 PowerPoint, but I do want to talk about a couple of key takeaways, one of which is just several of the vocabulary terms that we hit on today and what they meant, what they're important for. So the chromosomal basis of inherited disorders involves chromosomal theory, which identifies chromosomes as the genetic material responsible for what we now know as Mendelian inheritance, the process of inheritance, and genetic linkage, which explains that each chromosome can carry many linked genes, and this explains how individuals can have so many more traits than they have chromosomes. So back to the vocabulary terms, today we learned that a karyotype is the number and appearance of chromosomes, and it includes their length, the banding pattern, and their centromere position. So it's all about what the chromosomes look like. Non-disjunction is a process that occurs when homologous chromosome pairs or sister chromatids, which look the same and obviously are connected, when they fail to separate during the process of meiosis. An aneuploid is someone who is born with an error in chromosome number, for example, someone with Down syndrome, like we discussed. A chromosomal segment may duplicate or delete itself. Duplications and deletions often produce offspring that can survive but exhibit abnormalities. And a translocation occurs when a chromosome segment dissociates or breaks off from its original chromosome and reattaches to a different non-homologous chromosome. And in that process, we don't gain or lose any genetic information. It's just a process that happens and often leads to more genetic diversity. So once again, this is the end of our Chapter 13 PowerPoint. If you have any questions about the material, please contact me via email. And if not, move on to your next PowerPoint for this module. Thank you.